Hi, this is the first of two videos on translation as we continue our discussion of gene expression, which is turning specific genes on, and translation, specifically the act of making protein out of RNA. We'll learn that when we read the mRNA, the reading frame is crucial, that codons of 3 nucleotides specify amino acids. We'll be introduced to tRNA molecules, which are the adapter molecule between the nucleic acid code and the protein code. And we'll emphasize that proteins are synthesized in the amino to carboxy direction, also written as N to C. In the second video, we'll be introduced to the fact that the ribosome is the molecular machine, that it consists of many proteins and RNA molecules, that is responsible for making new proteins, and it's a quite remarkable machine. And we'll learn the 4-step cycle of making proteins from amino acids. In this slide, we want to emphasize a major difference between eukaryotes and prokaryotes. For a eukaryote to make proteins, DNA is transcribed into RNA here in the nucleus, depicted as a white circle. The RNA is spliced and capped. It will be exported out of the nucleus into the cytosol, which is where the protein is made from the mRNA. Therefore, there is a separation of the place where the RNA is made and the place where the protein is made. Prokaryotes don't have a nucleus, so DNA is made to RNA, and the protein is made from RNA in the very same place, and can even happen at the same time. While the RNA polymerase is making an mRNA molecule at the end part of the molecule, the ribosomes have already hopped on the beginning of the mRNA and started to make protein. Here we will explore what we refer to as the reading frame. This slide shows the relationship between one mRNA sequence. It is the same sequence here, here, and here. In the amino acid sequence, it can code for, depending on how we group them 3 at a time. It is how we group the nucleotides that is called the reading frame. The first thing you'll notice is that there are 3 nucleotides CUC, which, when they are grouped together, code for a single amino acid leucine. So the first thing you might ask is, why does it take 3 nucleotides to code for amino acid? You could even pause the video for a bit and think about it. The key thing to think about is that there are 4 nucleotides which need to code for 20 amino acids. Okay, I hope you gave that some thought. The answer is that 3 is the smallest number of nucleotides which can be used. If you only used one nucleotide, well, there's 4 different nucleotides that you could use, and you could only code for 4 different amino acids. So if you use 2 nucleotides, you could have 4 that go in the first position, and you could have 4 in the second position. That's a total of 16 different combinations. You could only code for 16 amino acids. It's only by using the third nucleotide, in which case you have 4 times 4 times 4, or 64 different possibilities, that you have enough coding potential to code uniquely for 20 amino acids. The second important thing to notice is the way you group the 3 nucleotides is crucial for specifying which amino acid gets incorporated. In this case, we're grouping the first 3 nucleotides. In the second case, we are grouping nucleotides 2, 3, and 4. In the third case, we're grouping nucleotides 3, 4, and 5. They all code for different amino acids: leucine, serine, glutamine. Normally, the cell, when presented with a sequence, would have to figure out what the reading frame is. We'll see later in the course some of the tricks the cell has for doing that, or computational approaches for doing that if you are analyzing a sequence. For this class, for now, you can always assume that when you are given an mRNA sequence and you have to convert it to a protein, you can always assume that the first 3 nucleotides are a codon, and then nucleotides 4, 5, and 6 is the second codon, etc. Here we see 64 different codons or arrangements of taking 3 nucleotides at a time. They've been stacked on top of the 20 amino acids depending on which one they code for. And there are 3 special codons that code for stop codons. This is the signal to the ribosome to stop making the protein. I'll just mention for your information that these 3 stop codons have special names. Sometimes you'll see it in the literature. They're called amber, ochre, and opal. I don't know which is which, and you don't need to know that either. All you need to know is that there are 3 stop codons. We can also see here that this is the 3-letter code for the various amino acids. Underneath, we have the one-letter code for amino acids. If you plan on going on in biological or biomedical research, then you might as well learn the single-letter codes, but you're not required to for this class. You do need to know the three-letter codes for this class. And as we've mentioned earlier, we need to know what class of amino acids they are. For instance, alanine, that's a hydrophobic amino acid. Arginine is a positively charged amino acid. Aspartic acid is a negatively charged amino acid, and so on. Now let's focus on alanine. There are 4 different ways to code for alanine: GCA, GCC, GCG, and GCU. You'll notice that in all cases the first 2 nucleotides are G and C. This is generally typical of the variation in the coding system. It's the 3rd position of the codon where one finds the most variability. For this reason, the 3rd position is called the wobble position, and it's generally less important for specifying an amino acid. The first 2 being the same doesn't always hold. For instance, there's 6 different ways of coding for serine. The first 2 nucleotides can be either AG or UC. Let's talk about the 6 different ways of coding for leucine. In principle, all 6 of these are interchangeable. The cell can use any of these at any time, and they will always specify leucine. However, in practice, the cell uses different codons for different proteins for different needs. There is a hidden layer of regulation. The most prominent example of this level of regulation is that some of these codons are used more frequently in highly abundant proteins. They have highly abundant tRNAs, and they are incorporated quickly into proteins. Some of them are used primarily for low abundant proteins, for which there are a low percentage of those particular tRNAs, and they get incorporated slowly. And that's one way that the system is optimized. We'll also see later in the course that once we know which ones are used for high abundant proteins, we can use this to make a fairly decent calculation about protein abundance based on its particular codon usage. Okay, I think this is the clearest way of depicting the 64 different codon combinations, but it's not the most common presentation. So we will look at that on the next slide. Let's focus in on the way to make methionine, which is AUG. So here we see the first base is A, and here U is the second base, and so that means it's somewhere in this column. And G is the third base, and that means it's here. And there it is, methionine. It's the only way to code for that amino acid. By the way, we need to remember that for the next video. AUG is methionine. Also, I should mention you don't have to memorize this table for the course. We will provide it for tests, but you will have to know how to use it when decoding RNA sequences. On this slide, we're introduced to the tRNA molecule, which is in essence the adapter between the mRNA sequence and the protein, because each tRNA carries an amino acid at the end. tRNA nucleotides read, or base pair with, the nucleotides of the mRNA, and a group of 3 is called a codon. Since the tRNA is also an RNA molecule, it will be able to base pair with the mRNA just like any 2 RNAs would. There are 3 specific bases on the tRNA called the anticodon, and those are the 3 that base pair to the 3 nucleotides of the codon on the mRNA. These red lines correspond to hydrogen bonds between the bases. The fact that there are 2, 3, or 1 red lines here are not of any particular significance, although I suppose they might be trying to show that the wobble position, position number 3, is the weakest, but I'm not certain. The final point is that since there are 61 different codons that code for amino acids, there are 61 different tRNA molecules that read the different codons. There are only 20 amino acids, but there are 61 different tRNA molecules. So in this slide, we see a more realistic depiction of tRNA structure. You can see that it has a sort of cloverleaf type structure here when unfolded. And that's the purpose of this little picture. Here's the anticodon. The anticodon is GAA. This is the anticodon for the amino acid, which is located here, phenylalanine. There's a D loop in red and a T loop in yellow, and here is the actual structure. It looks pretty crazy, but you can see that there's a blue loop, a yellow loop, and a red loop, and the amino acid is in green. And you can see some regions where you can imagine approximately a helical structure starting to emerge, but it's a far cry from the very regular, by comparison, structure of DNA. And remember, it's just that one hydroxyl group on the sugar that's the molecular difference. Yet look what an important structural consequence it can have, at least for nucleic acids. That's always amazing to me. And here at the bottom, we have a partial sequence. You'll notice that there's a funny symbol here. A few RNA molecules and ribosomal RNAs have a special base, psi. It's referred to as pseudouridine. I just point that out, but you don't need to know that for the rest of the course. Now in this slide, I just want to show you how the tRNA reads the mRNA. So here's the mRNA 5' to 3'. We're reading the mRNA in this direction. I had to flip it over and invert it, and then you can see that the anticodon A base pairs with U, A base pairs with U, and G base pairs with C. So this is the way that the anticodon is base pairing with the mRNA molecule. And UUC does in fact code for phenylalanine, which we can look up on our handy lookup table. The process of connecting an amino acid to a tRNA molecule, a pure tRNA molecule, is done by enzymes, which we'll see on the next slide. It utilizes ATP to make an activated adenylated amino acid, and this portion is then cleaved off as it is connected to the tRNA molecule. And this energetically pays for this otherwise energetically unfavorable reaction. You can imagine that it's very important for these enzymes to be accurate at connecting only the proper amino acid to the tRNA. Otherwise, when you're reading the mRNA, you would bring the incorrect amino acid and incorporate it into the growing polypeptide chain. Here we see the type of enzyme responsible for this reaction. It's called a tRNA synthetase. Since there are 61 different tRNA molecules, there are 61 different enzyme tRNA synthetases. Even though they're only connecting 20 different amino acids, you have to have one for the specificity of each tRNA molecule. In this case, the amino acid tryptophan is being connected to a tRNA molecule. Tryptophan is coded for by UGG. Now in this slide, we see the important principle that proteins are synthesized from the amino end to the carboxy end. In this case, there are 3 amino acids, 1, 2, 3, and connected to a tRNA. A 4th amino acid is going to come in. It's going to attach to the carboxy end of the peptide chain. In doing so, it's going to cleave the connection to the existing tRNA molecule. tRNA molecule number 3 will be released, and now you have 4 amino acids connected to the 4th tRNA molecule. This is all happening inside the ribosome, but this is the fundamental picture. You get that proteins are synthesized in the amino to the carboxy direction. And in the next video, we'll meet the ribosome and see how this occurs. Thanks for listening.